Thank you. Thank you so much. Sorry, I just heard my echo. Thank you for the invitation to speak and for the great introduction. And I'm really sorry I can't be there in person. And I hope we can at least share some of the exciting methods and findings from our lab here with you today remotely. So I'm going to speak about long molecule footprints of cancer genome structure. I'm an advisor to Impact Bio, but no other disclosures. So there's been a lot of rumbling about the imminent $100 whole genome era, and from Illumina itself, who the Illumina patents are soon set to expire. They apparently have the technology ready, and then more recently, announcements from new sequencing companies like Ultima Genomics that have new technologies that um, offer sequencing at $1 per gigabase and claim to go even lower. So I think we're at the cusp of $100 or maybe an even $10 whole genome era. Now this is short reads, but the question really is, can we routinely handle whole genome sequencing data in the clinic, and not just deal with the alignment and variant calling, but interpretation? And how can whole genome sequencing clinically add value, and also convenience? Now, these two questions I'm not going to directly address, but they're in the back of my mind and sort of motivating this talk. As mentioned, I'm a molecular pathologist and really interested in the clinical applications of genomics. But really what we want to think about also is, what is the next era of large scale genome studies going to look like? And with this new sort of promise of lower sequencing cost, is it better to invest in larger studies of hundreds of thousands of tumors, or given also recent advances in long read and long molecule sequencing, should we invest in maybe smaller but still expansive studies of long read profiles, whole genome profiles? And really, what is the value add of longer molecule profiling, which these days is in the range of 100 to 10 to 100 kilobase reads. And that's knowing that there are also technologies that aren't like sequencing, like optical mapping, that can give very high physical coverage that's well under $1 per gigabase. And so we're interested in structural variation and more broadly cancer genome structure, including 3D structure. So obviously, rearrangements are responsible for some of the most famous targetable alterations in cancer. But we're also interested in how these variants can perturb the non-coding genome, including 3D structure and enhancer hijacking. But also interested in thinking about how mutational processes shape genome structure, and how we can look backwards from the patterns we see in cancer to understand these mutational processes, some of which may be active even prior to the onset of cancer. And so the general intuition in genomics is that we need long molecules, whether long reads or optical mapping, to detect structural variants or large variants. And maybe there's a sort of something that's cognitively pleasant in thinking about large variants and long molecules. But I really want to approach this question critically, as somebody who's very excited about the promise of long reads, but also who's really interested in solving the underlying questions. And so what I'm going to talk to you about is two stories. One is where we've developed new methods and really applying them as far as we can to push the boundaries of short reads with a data structure and algorithmic paradigm built around something we call genome graphs. And then the second story is a vignette on how we use long molecules to understand structural variant patterns in homologous recombination deficient tumors. Sorry. Okay, so genome graphs. Oh yes, nice, animation works. So this is a user interface that we designed with the engineer in the lab, Haralampos Xanthopoulos, to allow us to interactively browse these structures called genome graphs, which are like copy profiles, but the graph structure arises from the fact that the nodes or the intervals in these graphs are connected to each other through rearrangement junctions, and both the nodes and the junctions are associated with a copy number. And when we browse these graphs, we want to see that those graphs actually mirror the observed binned copy number profiles that we see at a very high density in whole genomes. And the principle here is not a very complicated one, but it's also not a totally trivial one. And inferring these graphs is actually a pretty complicated task, but it's based on a simple principle, which is that every copy of every segment in a genome should have a left and a right neighbor. And that creates this kind of mass conservation principle along the genome that we can both exploit to get better copy profiles, but also to learn about the linear and possibly circular structure of rearranged alleles. And the algorithm that we've developed to infer these junction balanced genome graphs that obey this kind of conservation principle is called JABA. And we published on this a couple of years ago. And so one of the features that we can detect in these graphs is that junctions themselves have copy number. We think this is an important biological signal. Of course, that's familiar to those who do FISH, but it's a little bit less intuitive for those of us who study cancer genomes and are only often concerned with the copy number of rearranged segments. So once we infer these graphs, it actually becomes pretty easy to diagnose or detect complex structural variant patterns like these previously identified event classes like chromothripsis or chromoplexy or templated insertion chains. But we can also use these patterns in these graphs to infer novel classes of structural variants, like this pattern called rigma, which is a chasm of clustered deletions that we described in our paper a couple of years ago, which we can identify through sort of statistical analysis of many thousands of tumors. And these rigma occur in large, late replicating genes, and primarily in GI tumors like Barrett's esophagus and esophageal adenocarcinoma. And it's we're very encouraged to see others in the field using our tools. For example, this beautiful paper from Christina Curtis, which is a preprint where they modeled p53 deficient gastric organoids and showed the gradual evolution of these rigma, which is a sort of prediction from our study in this very controlled experimental system. So one of the sort of natural ways you can use these short read sequencing derived genome graphs is to try to infer the linear haplotypes in combination with long molecule whole genome sequencing. So this is linked read sequencing, which is barcoded short reads that are barcoded with their long molecule of origin. And this used to be an assay from 10x. Now others are offering a similar product, and using these long molecules, which are in the range of 20 to 40 kb, you can deconvolve these graphs into their constituent haplotypes. And that problem is ambiguous if you're only using short reads, but with long reads, you can actually phase the junctions and see which of them occur in the same linear allele. And there's even more power to do that. And again, beginning with our short read genome graph, where we have a very precise notion of dosage on both the junctions and the segments, we can use optical mapping, which is the sub-megabase molecules that you can physically map for quite cheap. And you can phase these junctions even when they're very far apart using these long molecules that are shown at the top. And that can help us resolve, in certain cases, rigma that we may have missed in short read whole genome sequencing. So do we need long molecules to detect large variants? And what fraction of large SVs are we missing with short reads? We wanted to approach this problem critically. And one way to think about it is something that to me was actually surprising, was to examine really the distribution of short read mappability of the human genome. Now, it's known that about half of the human whole genome actually comprises repeats, but a large number of these are pretty old repeats and are fully mappable. So in fact, if we look at base level mappability in the genome, 74% of the genome is base mappable. Actually, 87% of it is base mappable, but a fraction of 74% of it is base mappable, but a large fraction of these base unmappable bases are actually copy number mappable. That means that we can actually detect a very reliable copy number signal in these regions. You can see the binned read depth at the top of this diploid sample, but these unmappable bases are sprinkled at the rate of sort of 5 to 10% across these otherwise copy number mappable regions. And then the remainder of the genome is these no-go zones, which are almost 100% mappable. This is about 13% of the genome, where we can't get a reliable read depth signal. So already 87% of the genome technically should be accessible to short reads. So how does Java deal with missing rearrangements? Let's say we see these two tumor haplotypes, and that dark segment there at the top represents an unmappable region. Well, we're not going to detect that junction associated with the top haplotype, but we will detect the junction associated with the bottom one. So that'll give us a read depth profile in an otherwise CN mappable region that we can parse with Java. And by minimizing the residual of the read depth, which is this optimization problem that we solve, we can try to find the graph that most closely matches this rearrangement profile. Well, when we are missing rearrangements, Java is actually robust to these, because while we apply this junction balance criteria to make sure that every copy change is represented by a junction, we allow for these missing sites of missing dosage called loose ends, which are a slack penalty or a sort of regularization parameter in the Java model. And so using that, we can build our graph models, which will have junctions at sites of copy change that are associated with the rearrangement, but then we'll also have these loose ends, which look like these dangling loose ends at the bottom graph. So since our 2020 paper, we've improved Java significantly by applying these several improvements, which allow us to numerically converge this often difficult optimization problem, and also in helping model the inherent biases that are present in the genome when we measure read depth. And this is applying a method that we have in a preprint that was actually stuck for 9 months in genome biology in review before we got it back, saying that it was too boring. But this has been really a workhorse of our method and has really improved our performance. So we designed a really tough simulation benchmark to show to see how Java was performing, comparing it to other methods that are state of the art, as well as our previous approach. And what we found was that Java had extremely high recall. And this is where we put rearrangements both in unmappable and mappable regions, and really simulated the biases that are seen in real read depth data. So these are very gnarly benchmarks in impure tumors. And we found that our method had a very high recall and precision that was actually even higher than our previous Java version 0.1 method. So what are the insights that we gained from applying Java version 1? Well, one insight is that most rearrangements in cancer are actually unbalanced, and we can detect both balanced and unbalanced rearrangements. Balanced SVs are present in about 10% of all the detected junctions in these CN mappable regions, but most rearrangements are unbalanced. And we see both balanced and unbalanced loose ends. And you can see those as represented by these trailing blue edges on the right side of the plot. And you can see that most loose ends are unbalanced, but we can also detect balanced loose ends. And these most intuitively represent sites of missing dosage in the genome. So when we zoom in on these, and this is work led by a very talented MD PhD student in the lab advising it. So it's unpublished work. When we look at these loose ends, we can think of them in terms of a canonical coordinate, where in an unbalanced case, the loose end, the 5 to 3 direction is facing the side of copy number loss. And we can see that these loose ends are associated, as we would expect, with repetitive unmappable sequences on the actual reference locus that we're mapping to. And they're also associated with unmappable or repetitive or foreign sequences on the distal side of that rearrangement. And so these are true sites of rearrangement that we believe are missing from these genomes. And we can classify these loose ends based on the fact, can we assemble a de novo assemble a contig from the reads that are originating at that locus of missing dosage. And if we can assemble such a contig, we call that, sometimes we can find that that's actually uniquely mappable to a human sequence. It's just a junction that we missed with our initial caller. But in many cases, these are ambiguously mapped sequences that go to a line element, that go that connect a mappable sequence, usually a mappable sequence, to an unmappable repeat or a foreign viral sequence. And then in certain cases, we can't identify any distal mapping there, and we call those partially mapped loose ends. So quantifying these as a function of our total break ends, we find that these partially and ambiguously mapped loose ends represent less than 10% of all break ends in CN mappable regions. So this is quite a surprising result, because most it's the intuition in the field is that we are missing most structural variants. So we wanted to with short read. So we wanted to test this using long molecule profiling. We generated over 40 profiles of both linked read and short read whole genome sequences, and we compared the linked read derived break ends that we detected with those that we inferred in short read sequencing with Java, and we indeed verified that we are capturing the majority of SVs as either a loose end or break end in these CN mappable regions. So there's more to this story, but the key features here are that we can identify we can model the physical coupling between rearrangement and dosage in genome graphs. And that actually not only allows us to discover and classify complex SVs, but gives us a copy number caller that we think is the state of the art in the field. And finally, using the loose ends in these graphs, we can identify foci of unaccounted dosage and estimate the number of missing SVs in short read cancer whole genomes, and find the surprising result that we're missing fewer than 10% of large somatic SVs. So quickly, I want to pivot to our another story where we used long molecule sequencing to understand homologous recombination deficiency. So this is a disorder of the double-stranded break repair that leads to inherited cancer syndromes, but is thought to be also present sporadically in many cancers, and it is thought to be a big source of structural genomic instability, because homologous recombination is a main way that that normal cells repair double-stranded breaks, most commonly those that occur during DNA replication and in S phase. So there are signatures of these genomic scars that are detectable through methods developed by Sarima Zainal and colleagues. There's a bit of a paradox about them, because they mostly rely on very small variants. Now, like I said, HRD is thought to be a disease of structural genomic instability. So these classifiers mostly rely on SNVs, signature 3, or these microhomology-based indels, and this very non-specific signal of loss of heterozygosity, or these very local variant patterns of non-clustered tandem duplications that occur in BRCA1 deficient tumors or short deletions that occur in BRCA2. So we wanted to ask whether HRD tumors harbor distinct complex and large-scale patterns of double-stranded breakage, and can we use this to understand backup repair pathways and better classify HR deficiencies? So we generated a cohort of we collected a cohort of over 2,400 whole genome sequence tumors that we analyzed with Java, and first asked, since we can detect these various classes of complex SVs, can we find a consistent difference between BRCA1 and 2 tumors, particularly a class of variants that is uniquely seen in HR deficient tumors and not seen in wild type? And surprisingly, we found that actually these two classes of tumors were quite similar with respect to every complex SV class that we looked at, with the exception of these templated insertion chains where we saw some enrichment in BRCA1 and 2 deficiency. So while BRCA1, 2 deficient tumors do have these complex SVs, wild type tumors are also usually quite rearranged. So we wanted to take a little step back and think more broadly about complex structural variation, and particularly understand whether there were patterns that we could uniquely find in BRCA1/2 deficient tumors that we couldn't see in wild type. And complex SVs really represent are really derived from clustered break ends. Now, even in simple structural variants, reciprocal translocations and inversions, we see break ends that are clustered, and often a single break end where both ends are going to a different location. But most even simple structural variants don't have this exact reciprocality. In fact, they have approximate reciprocality, where you have these short segments called gaps that we call gaps that separate the two break ends. And those gaps can be in one of two orientations, where the break ends are connected to that intervening segment, which is we call positive polarity, or those segments are facing away, which we call negative polarity. And based on these different orientations, you can actually have different interpretations. For example, on the right, you can have either a reciprocal translocation with a duplicated segment, or you can get a templated insertion, which is a copy-paste event. While on the left, you have two reciprocal translocations. This is when these two loci are on different chromosomes. And when you see a gap, there is an intervening loss. So applying this notion, kind of enhanced generalized notion of reciprocality across our pan-cancer data set, we found that we could represent many classes of both simple and complex SVs as various topologies of these gaps connected to each other in various sort of reciprocal orientations. And this included various classes of templated insertion events or chromoplexes. And surprisingly, we found that BRCA1 and 2 deficient tumors were still very similar to wild type with respect to these more complicated topologies, but we found a very significant enrichment in these with respect to these paired topologies. So specifically, we found that BRCA1 deficient tumors had these enrichment of these paired reciprocal events, where BRCA1 had these reciprocal duplications, where there were two gaps of positive polarity connected to each other through rearrangement. BRCA2 deficient tumors had these reciprocal deletions that were in the bottom left quadrant here, where you have two negative polarity segments connected to each other, and both had these mixed polarity events called reciprocal, that we call RDEL dupes. So looking at a leave-out set of tumors, we found that indeed these R dupes were enriched in BRCA1 deficiency, RDELs were enriched in BRCA2 deficiency, and these RDEL dupes were enriched in both BRCA1 and 2 and almost absent from wild type. So to investigate the phase of these variants, we generated a large data set across 98 samples from BRCA1 and 2 deficient tumors, sequenced a very deep physical coverage, 170x, with a median molecule depth of 21 kb. And we validated these associations of these events, but what we wanted to understand was the topology and the actual structure of these rearranged loci. So these reciprocal deletions are essentially, when they're interchromosomal, are balanced translocations where the intervening segment is deleted, and they have only one possible outcome. And so what we expected with our linked read data was to see this sort of pattern where the heat map there shows the patterns of barcode sharing, sort of similar to a high C map expected with this kind of trans phase. And indeed, that's what we found with these reciprocal deletions. But reciprocal duplications, what we actually expect to see are a few different, very distinct orientations. And one class of these would be a copy and paste event, where either the B segment in one locus is copy and pasted into the second locus, or vice versa, which would give you this kind of striped pattern with respect to the linked read barcodes. Or alternatively, you could see a translocation where both derivative chromosomes have duplicated material at this gap, and that would give you the sort of pattern that we see above. And while we saw many cis reciprocal duplications, we actually saw quite a few of these trans orientations that represented either inversions or approximately balanced translocations with duplicated material, at about half the rate of where we saw these cis copy and paste events. And looking into the orientation of these rearrangements, we saw that the cis and the trans configurations have very distinct features, where in the cis, we saw a very small segment that was inserted in between two much longer segments, and that's a copy and paste scenario. And in the trans orientation, we saw two large segments that were fused to each other. And so I'm just going to fast forward here because I'm running out of time, but we can actually, so what's the significance of this? I think they fit, the way we can interpret these events is actually in a continuum variation that we can actually link backwards to the tandem duplications, which are classically, or have been for a long time, associated with BRCA1 deficiency. And we think that these reciprocal pairs arise from the same replication restart mechanism involving a non-homology driven form of breakage-induced replication. And the significance of these cis and trans events is that the cis form is very similar to these tandem duplications that involves just a simple copy and paste event. But this trans configuration can give rise, in combination with missegregation, to the LOH events that we see that are long associated with HRD. So in summary, while BRCA1/2 deficient tumors are quite similar to wild type, this enrichment of reciprocal pairs provides a source of long-range trans structural variants. And we can link this to a shared mechanism. And what I didn't show here is we can use that to actually create a predictor of HR deficiency that outperforms the state-of-the-art. So in summary, these genome graphs are really a powerful paradigm to understand complex structural variants from both short and long reads. And they help us estimate how complete are our short read reconstructions in the context of anticipating the advent of long molecule technologies. And then we can apply long molecule whole genome sequencing to actually phase multi-junction rearrangements and identify these novel cis and trans patterns in BRCA1/2 deficiency that we think has a mechanistic and also a potentially clinical implications. I want to thank the folks involved in this work, particularly Kevin and Zining, and our collaborators at MSKCC, especially Simon Powell and George Rice-Filio, who we collaborated on this, and our funding sources. I'm sorry for going over time. Hopefully we have some time for questions. Thank you.
Thank you very much, Marcin, for the excellent talk. Very informative information. So we'll dedicate the next 4 or 5 minutes for our Q&A for the sake of time. Please feel free to go to the mic if you have any question. Yes,
thank you so much, Marcin. This was an amazing talk, and thanks for the presentation at the CGC. So my question, and I apologize if I missed it, but for extracting the CNVs and the structural variants from short read, what is the average, or what is the minimum depth of coverage that we need to have?
So in our analysis, we've oh, sorry, just the feedback. In our analysis, we've been looking at genomes that are about 40 to 50x with purity greater than 50%. But I think for clinical grade whole genome sequencing, I think what we need is probably something closer to 80x in tumors. And there's two goals of the analysis. One is to accurately detect read depth at both sort of genome-wide and at heterozygous SNPs. And the second is to detect these junctions. And then what JABA does is help put those together. But it's robust enough to missing rearrangements that I think we can deal with both impurity and lower read depth. But there are some who recommend doing 120x, like the Hartwig Medical Foundation, which has been pioneering clinical whole genome sequencing in the Netherlands. routinely will sequence to over 100x.
Thank you very much. We have one more question from our in-person audience. Jade, please.
Yes, and this is a question both for Marilena and Marcin. Thank you, Marcin, for that very comprehensive explanation. Given that whole genome sequencing is now in the clinical laboratory at New York Genome Center, has JABA1 been incorporated in that pipeline?
Yeah, this is an excellent question. So Marcin, please go ahead. Actually, I had a similar question, so you can go ahead and reply.
Right. So Java version 1 is still it's a manuscript in revision. So the short answer is no, it hasn't yet been incorporated, and for good reason. The regulatory sort of process that you need in order to get both New York State and CLIA approval, goes all the way down to specifying the exact pipeline for every single variant class. So I think it is our goal to incorporate Java into the clinical workflow. It's not there yet. I think the state of the art currently is a consensus approach that uses classic SV callers that don't use this graph approach.